Have you ever considered the sheer audacity of existence? The intricate, almost unbelievable machinery that hums within each of us, allowing us to think, to feel, to simply be. It all comes down to a molecule, a breathtakingly complex molecule. Look around you. At the people you love, the trees swaying in the breeze, the vastness of the sky. Everything living shares this same, fundamental code. But it's a code so hidden, so deeply woven into the fabric of life, that we didn't fully understand it for decades. Imagine taking just one cell from your body, gently unraveling its core and stretching it out. That single strand of DNA, if laid end to end, would be taller than you. Now consider this. The human body contains approximately 50 trillion cells. Stretch all that DNA out, and it would reach the sun not once but 600 times. 600 times. Does that begin to hint at the immensity contained within us? You probably know it by its initials, DNA. Deoxyribonucleic acid. It's the blueprint, the instruction manual, the very essence of who you are. A 6 billion letter code dictating everything from the color of your eyes to your predisposition for certain traits. And it's not just your code. It's the code for nearly every living organism on this planet. A universal language spoken by every plant, every animal, every bacterium. If you're human, every single one of your cells, excluding red blood cells, contains 46 chromosomes. Each chromosome is a tightly wound package of DNA, nestled safely within the cell's nucleus. DNA belongs to a family of molecules called nucleic acids, alongside its cousin, RNA. Remember those fundamental building blocks of life: carbohydrates, lipids, and proteins. Nucleic acids are the fourth major group, and arguably the most crucial. They bear the weight of heredity, the legacy of generations. Like any complex structure, DNA isn't just one piece. It's a polymer, meaning it's built from many repeating smaller units. In the case of DNA, those units are called nucleotides. Think of them as the microscopic Lego bricks that assemble into something magnificent. To build a DNA molecule, you need 3 core components for each nucleotide: a 5-carbon sugar molecule, a phosphate group, and one of 4 nitrogenous bases. The name itself, deoxyribonucleic acid, gives away the first ingredient, deoxyribose, the sugar. But the real magic, the genetic information that defines you, lies within those 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. These aren't just random letters, they are the alphabet of life. The specific order, the sequence, is everything. AGGTCCATJ is a completely different message than TTCAGTCJ. Even a single change can have profound consequences. Now imagine this. DNA doesn't exist as a single strand, but as a pair, intertwined like lovers in a graceful dance. It's the famous double helix, a microscopic spiral staircase. And like any sturdy structure, it needs a strong support system. That support comes from the sugar and phosphate molecules, forming the backbone of each strand. These backbones run in opposite directions, one appearing upside down relative to the other. A subtle detail, but crucial to understanding how everything fits together. This directionality is described as 5' and 3', referencing the carbon atoms on the sugar molecule. Picture the deoxyribose sugar, with an arrow pointing from the 3rd carbon to the 5th. One strand starts at the top with a phosphate connected to the 5th carbon, ending with a free end at the third. The other strand is its mirror image, beginning with a free end at the third carbon and ending with a phosphate at the fifth. It's a beautifully elegant asymmetry. Let's build our own double helix. The two strands are connected by the nitrogenous bases, held together by relatively weak hydrogen bonds. But here's the catch: they aren't random pairings. Adenine always pairs with thymine. Guanine always pairs with cytosine. These are the rules of the game, the grammatical structure of the genetic code. Guanine-cytosine pairs form 3 hydrogen bonds, making them slightly stronger than adenine-thymine pairs, which have only 2. It's this precise sequence of base pairs that makes you, you. Chromosome 1, the largest in our genome, contains a staggering 247 million base pairs. If you were to print out those letters, it would fill a book of roughly 200,000 pages. And each of your cells has 46 such DNA molecules crammed into its nucleus. To contain your entire genome, you'd need approximately 10,000 of those 200,000-page books. A library of you contained within every cell. Quick test: can you predict the complementary strand? Let's say we have 5' AGGTCCG to 3'. What's the corresponding sequence? The answer, of course, is 3' TCCAGGC5'. Simple, right? Because each base has only one partner, deciphering one strand reveals the other. Now let's bring in RNA. It's similar to DNA, a sugar-phosphate backbone with nucleotide bases attached. But there are key differences. First, RNA is usually single-stranded, lacking that elegant double helix. Second, its sugar is ribose, with an extra oxygen atom. And third, RNA doesn't contain thymine. Instead, it uses uracil, which also pairs with adenine. RNA plays a critical role in protein production, and crucially, in replicating DNA. But before we dive into replication, we need to acknowledge the incredible minds behind these discoveries. It wasn't a single eureka moment, but a century-long journey, a collaborative effort of brilliant scientists from around the globe. James Watson and Francis Crick are often credited with discovering DNA, but that's a simplification. They didn't discover DNA, they elucidated its structure. The story begins in 1869 with Friedrich Miescher, a Swiss biologist who, in a rather gruesome manner, obtained white blood cells from used hospital bandages. He extracted a strange, phosphorus-rich substance from the cell nuclei, calling it nuclein. He knew it was new, but its purpose remained a mystery. Nearly a century later, Rosalind Franklin, a young biophysicist in London, brought us closer to the truth. Using X-ray diffraction, she captured images of DNA that revealed its helical structure and showed that the sugar-phosphate backbone was on the outside. Why isn't her name more widely known? Sadly, Franklin generously shared her data with Watson and Crick. Her crucial images were shown to them without her knowledge, influencing their model building. She published her findings, but after Watson and Crick had already published theirs, offering only a brief acknowledgement of her contribution. Tragically, Franklin died of ovarian cancer at the age of 37. The X-ray technology she used exposed her to dangerous levels of radiation, and she rarely took precautions. The Nobel Prize can't be awarded posthumously, and many believe she would have shared the 1962 prize had she lived. Her story is a stark reminder of the often overlooked contributions of women in science. Now that we understand the structure, let's explore how DNA copies itself. Cells constantly divide, and each new cell needs a complete set of instructions. This process, called replication, is remarkably efficient. Our cells can copy this entire genetic library in just a few hours. It's like creating 10,000 copies of a massive book in the blink of an eye. The process begins with an enzyme called helicase, unwinding the double helix and separating the two strands. The point where the separation starts is called the replication fork. These unwound strands now serve as templates for building new complementary strands. But there's a catch. Remember that the two strands run in opposite directions. This creates a challenge. One strand, the leading strand, is relatively easy to copy. An enzyme called DNA polymerase simply adds matching nucleotides to the strand, continuously building the new DNA. But before it can start, it needs a small primer, a short sequence of RNA created by another enzyme called RNA primase to get things going. Once the primer is in place, DNA polymerase smoothly adds nucleotides along the strand. The other strand, the lagging strand, is more complicated. Because it runs in the opposite direction, DNA polymerase can only work backwards, creating short fragments called Okazaki fragments. RNA primase lays down multiple primers along the lagging strand, and DNA polymerase fills in the gaps between them. Finally, another DNA polymerase replaces the RNA primers with DNA, and an enzyme called DNA ligase glues the fragments together. DNA replication isn't perfect. Errors occur about once in every 10 billion nucleotides. But our cells have a built-in proofreading system. DNA polymerase can detect and correct mismatched bases, ensuring the integrity of the genetic code. Considering the sheer scale of the operation, millions of miles of DNA within each of us, it's astounding that mistakes are so rare. This, my friends, is why scientists rightfully call DNA the most celebrated molecule of all time. It's a testament to the elegance, the complexity, and the sheer wonder of life itself. Take a moment to appreciate the miracle unfolding within you right now. Revisit this episode, delve deeper into these concepts, and marvel at the intricate world of DNA. And as you prepare for next week, remember this: understanding the structure of DNA is only the first step. The real magic lies in how this code directs the creation of you. Thank you to the incredible team at Crash Course who brought this episode to life. Explore the resources below to revisit key concepts or delve deeper into specific topics. Share your questions and thoughts in the comments or connect with us on Facebook or Twitter. The journey of discovery continues.